Dear students, in this module, we're going to look at the process of protein folding. You know that Levinthal's paradox states that the time required to compute the structure of a single protein is more than the time this universe has existed. So, we're left with no option but to actually decipher how a protein folds itself, because it does that in a very short span of time. Naturally, if it takes that much time, that is a very long time to compute so many possibilities, then it is nearly impossible. We are only left with a single option, and that is to understand the folding process in nature. The folding takes place as a result of 4 different forces. Number 1, electrostatic interactions that occur between charged portion of the proteins, the van der Waals forces, the attraction between the masses which are very weak, hydrogen bonds that are formed, and the hydrophobic interactions. Hydrophobic interactions would be the push the amino acids they achieve after rejecting to take water. So these interactions, they come together and give rise to the folded proteins. Let's take an example. So if you have a simple polypeptide, amino acids chain, then it takes a preliminary folded form as shown here, and eventually the entire protein gets folded into a functional protein. Now, all of these 4 interactions, they may be taking place at the same time at several locations within the protein. For instance, there may be some hydrogen bonds that would have been made, right? There would be some electrostatic interaction that is going on, and there will of course be van der Waals forces that are acting, and all of this put together will result in the folded protein. So essentially, if we were to understand all of these 4 different factors, then we can arrive at the folded structure ourselves as well. So Anfinson's experiment, it showed us a very interesting insight to this folding process. Here, he took a ribonuclease in its native form, in its folded form, and then he denatured it, and it got converted into a linear chain. So essentially, this is unfolding a protein. So once he was able to achieve this, then this unfolded protein, it lost its function. So this protein was no longer able to perform the functions that it was able to perform in its folded shape. Next, Anfinson folded the protein again by allowing it to mix with water, and the protein took its native form again. You can see that the protein is in its folded form. Amazingly, this protein started to function as it functioned before the denaturing process. The resumption of the function of this protein actually showed us that the information that is there and required for the folding of a protein is actually present within the amino acid sequence. So the 4 different interactions that I have talked about are essentially going to take part on behalf of the amino acid sequence. So if you have a structure that is if you have a protein in its folded form and you denature it, and if you allow it to fold again, then it can totally achieve its folded form and function once again. So this was an amazing finding, and this guy also got a Nobel Prize for his work. The take-home message would be that the protein achieves the folded form again after considering the stability, the thermodynamic free energy that is there and by minimizing it. So once it has minimized the free energy, then the protein is essentially folded back and it can function again.